Alright guys, today we're going to cover everything that you need to know about enzymes for AP and introductory college bio classes. So enzymes are proteins that speed up chemical reactions. If you don't already know, proteins are molecules that are commonly found in a cell that are a bunch of amino acids that have linked together into a chain, and these amino acids attract or repel each other to make a certain shape. Once this chain is in a certain shape, that is a protein. And these proteins can have a variety of functions, one of them being an enzyme or something that speeds up a chemical reaction. And something really important for proteins is the way they're formed is extremely important because their shape is everything for how they function. We can see here in the shape of the enzyme, we have two little sites to accept what's going to be put together in a chemical reaction, which we'll see in a bit. Shape determines function. Enzymes are proteins that speed up chemical reactions. So we've covered the protein part, but what about the speeding up? How does an enzyme speed up a chemical reaction? Let's start with an example. Let's say there was a chemical reaction where molecule A comes into contact with molecule B, and they form molecule C. And I just have my molecules A and B just out and about, and they're just moving, right? But in order for this reaction to happen, I need molecule A to come into contact with molecule B. And with the way they're moving now, they could be moving around a lot without ever coming into contact. All of this movement, all of this energy expended into moving these molecules around so they hopefully hit each other, can be really wasteful. It can be too much energy spent in hopes that molecules A and B come into contact. So what an enzyme does, or one of the ways that they speed up chemical reactions, is that they have sites that are specific for and are attractive to molecules A and molecules B, and then they just bring molecules A and B together, thereby saving a ton of energy. Molecule A and B are substrates, and what's released from the active site once the reaction has occurred is molecule C. So that's how enzymes save a ton of time and energy. So what an enzyme does, an important phrase to remember, is that they lower the activation energy, or the energy needed to get a reaction started. The activation energy in our situation was all the energy needed to move these molecules around so that they hopefully hit each other. With something like an enzyme that just brings them together, we have greatly lowered that activation energy. So a question that I encountered a lot when taking the course was, can enzymes change reactions from exergonic to endergonic, or maybe the reverse? Before we answer the question, we have to first look at what an exergonic and endergonic reaction are. So here we have both of our reactions. In an exergonic reaction, we have substrates A and B, and they come together to create product C. And upon the creation of product C, energy is released. Overall, we have released energy with this reaction. In an endergonic reaction, energy has to be put in with substrates A and B to create product C, so energy has been absorbed throughout this reaction. Exergonic means energy is released after the reaction, and endergonic means energy had to be put into the reaction. So they're opposites in that sense. So now we're going to take a look at an exergonic reaction without an enzyme, then add an enzyme, and see if we can make it into an endergonic reaction. We're going to be showing the reaction over here, and we're going to be putting on a chart the energy and the reaction progress. So to start off, at the very beginning of the reaction, we have our energy at a certain level. These molecules are moving around as normal at some certain temperature, and we have our molecules A and B. This is our system in which the reaction is going to occur, this box right here. Then what happens is that I have to put in a bit more energy for these molecules to move around enough to eventually hit each other. This is called the activation energy. It's the energy I have to put in to get the reaction started and get A and B to hit each other. Then, because we're looking at an exergonic reaction, once product C is created, energy leaves the system, leaves our box, so that means this box has less energy than when we started. So that is the chart for our exergonic reaction. Now let's add in an enzyme and see if we can change it from exergonic to endergonic. So we started off like normal. We have our molecules moving at a certain pace, at a certain temperature, at this certain energy right here, right at the beginning. Then our enzyme comes into play and greatly reduces the amount of movement or energy energy expenditure by A and B in order for them to come together. So that greatly reduces my activation energy, but it doesn't really change how A and B started or how they end up creating C and the energy that's released because C was created. So our energy beginning point is the same, our energy endpoint is the same, we release the same amount of energy, and C was created all the same. All that was changed was the activation energy, or the energy needed for A and B to come together. So because they're unchanged, we didn't actually change the reaction from exergonic to endergonic. And that's really important to remember. Enzymes cannot change reactions from exergonic to endergonic. They only lower the activation energy. So, quick review of what we said before we move on to the next step. Enzymes are proteins that speed up chemical reactions, but cannot change the start or end point of a reaction, meaning they cannot change a reaction from exergonic, energy releasing, to endergonic, energy absorbing, or the other way around. A term I heard often is biological catalyst. Biological because it's in a living thing in a cell, and what a catalyst is a molecule that helps speed up a reaction and does not get used up itself in the reaction, sort of like an assistance for that chemical reaction to get carried out. They are biological catalysts. Enzymes are proteins that are working to keep cells alive, and they don't get used up themselves in reactions. So they are super useful because a lot of the reactions that are needed to keep a cell alive would take way, way too long if they didn't have enzymes. Cells or life on Earth wouldn't be possible without enzymes because too much time and energy have to go into those reactions that keep us alive. So now let's take a closer look at the enzyme, sort of look at that structure. Here, in these two little spots perfect for molecules A and B to join, is the active site. The active site is where the enzyme accepts substrates. There are many different types of enzymes, and there are many different types because each type is specific to a substrate. So there's a specific reaction, and then there's a specific type of enzyme that only accepts certain substrates for that reaction. So these active sites are really specialized for the certain type of substrate involved in whatever chemical reaction the enzyme is supposed to assist. So all these spots around here, outside of this active site where the substrates are supposed to bind, if anything binds to any site outside of the active site, it is known as binding allosterically. We're going to see a lot of molecules that don't bind directly to the active site, and they have all bound allosterically. That's a really important vocab term. So a closer look at the enzyme, one of the most common ways enzymes accept substrates is via induced fit, which is where the substrate approaches the enzyme and the enzyme sort of melds its structure around the substrate in order to accept it. So we covered what they are, how they work, and some of their features. Another really important concept to learn about enzymes is things that can help or stop them. So first off, we have environmental conditions. Things like extreme pH, extreme temperatures, and chemicals can interfere with the bonding that's occurring or the attractions and repelling that's occurring between the amino acids to form the shape of the enzyme. If these attractions or bonds are interfered with, then that means the shape is interfered with, and an enzyme like any protein, shape is crucial for function. So that means the function of the enzyme is going to be gone. The structure is going to be all messed up, and it's not going to function anymore. The term for this loss of shape and this loss of function is known as denaturation, or an enzyme has been denatured when its structure has been ruined and it can't be used anymore. For those who don't know, pH is essentially the number of hydrogen ions nearby. So if there are a lot of hydrogen ions nearby, they could interfere with the attraction or repelling between, let's say, some polar amino acids. And if these hydrogen ions or the extreme temperature, all that heat, all the molecules moving and being ripped apart in terms of their attraction or their bond to each other, or possibly chemicals, if that structure is ruined, this can greatly interfere with the structure of the enzyme. So for every enzyme, there is a certain pH range and temperature range in which they function best. And outside of that range, they can denature. There are molecules that can assist in enzyme function, and they can be bound loosely or tightly. If this molecule is organic, it is known as a coenzyme, and if it is inorganic, it is known as a cofactor. What these molecules do is help assist the enzyme in functioning and doing its job. Organic means containing carbon and being used in living things. Inorganic molecules can be certainly be used in living things. It's just they don't contain carbon. They're typically metals. So when an enzyme is joined with these assisting molecules, they can form what is known as a holoenzyme. Up next, we have inhibitors. Inhibitors are molecules that can attach to an enzyme and cause it to not be able to do its job anymore. Here I'm showing that by pretending it's closed off that active site so no more substrates can join. Inhibitors can bind permanently or reversibly. If they bind permanently, they're using something like covalent bonding, or where electrons are being shared. And if it's reversible, that means it can be undone because it's just a mild, weak attraction. There are two types of inhibitors, competitive and non-competitive. If it is a competitive inhibitor, we can see here that it is binding directly to the active site. In other words, it's racing with the substrates to see who can reach the active site first, because when one of them reaches, that means the other one can't use the active site. Non-competitive inhibitors, we can see that it's not a race because the inhibitor can just bind allosterically or to somewhere other than the active site to prevent the enzyme from being able to take the substrate. So one thing to know about competitive inhibition is that if we add more substrate, the effect of the inhibitor can be reduced. However, this is not true for non-competitive inhibition. In non-competitive inhibition, because the inhibitor is binding allosterically, then the addition of more substrate wouldn't really affect anything because it's not stopping the inhibitor from reaching the enzyme. So next we have activators. Activators are, I like to think of it as just the opposite of inhibitors. They're molecules that can bind to an enzyme and help activate it so it can get started and assist in doing its function. So a question format that I saw a lot when taking this course was you would have a series of reactions, like reaction 1, reaction 2, reaction 3, and a product. And this is like a biological pathway in the cell. Each of these reactions have their enzymes. And for each of these enzymes, you'd have to predict what would happen if an activator or inhibitor were added. So, for example, if we added an activator to this enzyme, that would improve this enzyme's function and cause more of the product to be created. If we had an inhibitor over here, that means that this enzyme is inhibited, this reaction is reduced, meaning less product. And where it got a little bit trickier was, let's say an inhibitor for one enzyme was created from a reaction which was assisted by its own enzyme. If an inhibitor was added to this enzyme, that means that this reaction is reduced, meaning there's less of this product, which is an inhibitor for this. So less inhibitor over here means this enzyme is less inhibited, so this reaction can occur more, so then more product is being created. And it's kind of that pathway, calculation with pathways that I saw a little bit in some of those AP Bio questions relating to enzymes and biological pathways that showed that it's really important to have solid what an inhibitor and an activator does. So I would recommend being ready for that kind of question. To finish off, we have cooperativity. Some enzymes have multiple active sites. Here we can see this enzyme has 4 active sites. But they're not always all open. Sometimes these enzymes only have 1 active site open while the other active sites are closed. If a substrate binds to this 1 active site, it can sometimes cause all the active sites to open up. And this is what is cooperativity. And it is actually a form of allosteric activation, because in this situation, our substrate is actually also working as an activator, because it's activating the enzyme and causing it to open up all of these other active sites. Since this is a form of activation where the substrate acted as an activator and it bound to somewhere other than the active sites that got opened up, it is a form of allosteric activation. All right, so that should be everything that you need to know. Good luck on your test tomorrow.